hearing loss after spinal anesthesia, the use of hyperbaric oxygen therapy, HBOT, followed by Perispinal Etanercept, PSE, injection. Etanercept is a potent antagonist of TNF, a pleiotropic immune signaling molecule that is also a pivotal regulator of synaptic function. Excess TNF is centrally involved in the pathogenesis of a variety of inflammatory neurological disorders, including Alzheimer's disease, sciatica, traumatic brain injury and spinal cord injury. Perispinal Etanercept produces rapid improvement in both Alzheimer's disease and sciatica and in other forms of disc-related pain. Basic research and the observed clinical effects suggest that Etanercept has the surprising ability to penetrate into the cerebrospinal fluid after perispinal administration. Perispinal administration is a novel method of delivery designed to introduce this anti-TNF molecule into the bidirectional cerebrospinal venous system and the cerebrospinal fluid to facilitate its selective delivery to either spinal structures or the brain. Etanercept, trade name Enbrel, is a biopharmaceutical that treats autoimmune diseases by interfering with tumor necrosis factor, TNF, a soluble inflammatory cytokine, by acting as a TNF inhibitor. It has US FDA approval to treat rheumatoid arthritis, juvenile rheumatoid arthritis and psoriatic arthritis, plaque psoriasis and ankylosing spondylitis. TNF-alpha is the master regulator of the inflammatory, immune, response in many organ systems. Autoimmune diseases are caused by an overactive immune response. Etanercept has the potential to treat these diseases by inhibiting TNF-alpha. 1. Etanercept is a fusion protein produced by recombinant DNA. It fuses the TNF receptor to the constant end of the Ig1 antibody. First, the developers isolated the DNA sequence that codes the human gene for soluble TNF receptor 2, which is a receptor that binds to tumor necrosis factor alpha. Second, they isolated the DNA sequence that codes the human gene for the Fc end of immunoglobulin G1, Ig1. Third, they linked the DNA for TNF receptor 2 to the DNA for Ig1 Fc. Finally, they expressed the linked DNA to produce a protein that links the protein for TNF receptor 2 to the protein for Ig1 Fc. The prototypic fusion protein was first synthesized and shown to be highly active and unusually stable as a modality for blockade of TNF in vivo in the early 1990s by Bruce A. Beutler, an academic researcher then at the University of Texas Southwestern Medical Center at Dallas, and his colleagues. 234. These investigators also patented the protein. 5. Selling all rights to its use to ImmuneX, a biotechnology company that was acquired by Amgen in 2002. 6. It is a large molecule, with a molecular weight of 150 kDa, that binds to TNF-alpha and decreases its role in disorders involving excess inflammation in humans and other animals, including autoimmune diseases such as ankylosing spondylitis. 7. Juvenile rheumatoid arthritis, psoriasis, psoriatic arthritis, rheumatoid arthritis, and, potentially, in a variety of other disorders mediated by excess TNF-alpha.